Yeah, good morning friends. This is Vinoy Krishna and we are connecting today on the Vajiram platform to discuss on the Nobel Prize on Physiology or Medicine that has been announced for 2024 by the Royal Swedish Academy. Let us begin with a simple curiosity as to there are two Nobel Prizes as being shown on the screen at a distance of almost 18 years between them. One that was awarded in 2006 to a couple of Americans, Andrew Fire and Craig C. Mello. And then fast forward to 2024 when the Nobel Prize in Physiology has been awarded to again a couple of Americans, Victor Ambrose and Gary Ruvkun. Now what is such a connect between these two Nobel Prizes? People, as we go on to discuss the entire sequence of events, we will find that both these Nobel Prizes are largely talking about the ultimate similar or same applications. Let us begin with the present issue first. Mr. Victor Ambrose and Gary Ruvkun, these two Americans, they have been awarded the Nobel Prize for having decoded one of the biggest mysticism in the life's diversity as to why, when all of us as an individual are made up of trillions of cells which at the very basis are absolutely the same, and yet functionally they are quite diverse. Just to tell you, let us say, if you talk about the story of a human, it begins with the first cell called zygote, ultimately dividing multiple times to become a human made up of trillions of cells. Now, interestingly, all of these cells, like somewhat around 30 to 50 trillion cells that we are made up of, are the carbon copy of zygote. No difference with respect to the basic plan of life that they are endowed with. We're talking about the genetic material. This discourse is on the assumption that the aspirants at the moment are kind of largely aware about what do we mean by the genetic material and how all the informations for a particular organism and the various physical, physiological and behavioral characteristics are stored. Let us say if a human represents the trillions of the cells, within each cell we have the information of what the cell and the organism of which the cell is a part of has to behave like is all stored in the molecules called DNA. These DNA are packaged in the form of chromosomes, and we know that a human's cell has absolute 23 pairs of chromosome. Each chromosome having the helical structure called DNA, which is nothing but a chemical molecule, and on that DNA, the information of the different characteristics are stored in the sequence of letters or bases. 4 of them you might have heard about, A, G, C and T. Based on all the sequences possible, we can figure out what information that particular DNA holds. Now, the entire set of DNA that is present inside the cell together makes up the entire information that a cell has. And it is the same story for every cell of the body. Now that is called as genome. If we look into the genome, people, you will find that the genome or the total set of DNA does not have the entire region of it having the meaningful information with respect to the coding of proteins. So what we know now, that only some selective segments, what is being shown in the green here in this image, are the ones coding for proteins, and these segments are called as genes. The human genome, or all the 23 chromosomes together, have near about 20,000 genes in total, 20 to 25 at the max that we are presently estimating. Now, all the genes also, people, if you account for, you will find that they will explain for only 1.5% of the total genome, meaning the remaining 98.5%, what is being shown here as intergenic region in the DNA, are casually referred to as junk DNA, or to be more precise, called non-coding DNA regions. For a long part of the biological scientific history, we assumed that this is functionally meaningless, and hence the term was given as junk. But over the last few decades, the accumulation of informations have only added to the information that, okay, they might not be directly involved in the protein synthesis, but they have a massive role to play in regulating the process. So let us quickly again review what you already might know is the truth of life for every living organism within a cell. Within each cell of your body, inside the nucleus, the entire genetic material called DNA, having the sequences of letters and the information are present. Every time any particular gene of the available 20,000 is required to express itself, the information to be led to the product. It does not get involved directly, but it creates its carbon copy or the photocopy in exactly the same manner as you might have seen the printing on the clothes. People, this cast is more like the DNA, and what it is leaving is the same information on a structure, let us say the RNA. So the information of DNA copied in the form of RNA inside the cells. And that information of the RNA, people, is then read and decoded to produce the most versatile molecules within the cells that we have, or the proteins. The proteins are formed by reading the information of the RNA, and hence that RNA, you might be knowing, is popularly called messenger RNA because it has carried the message of the gene and is decoded, and that means you are converting the information of the RNA into a sequence of amino acids, what we call as proteins. Now, despite we having only 20,000 of genes, there are infinite number of proteins that could be created. When we discuss the Nobel Prize in chemistry, you will realize how today we are sitting at a point wherein we can even create or design new proteins for new applications, which is again going to lead to a lot number of applicational avenues. That's the Nobel Prize in chemistry, quite connected. We'll come to that later. But now, what we know is this is what is central to life. Every organism does that. The gene copied into RNA and then translated into the protein. Copying into the RNA is called the transcription, and converting it into the protein, because proteins are different molecules altogether, that process is called translation, converting one language into another. Now people, the biggest curiosity that we had, how come all the cells of the body having the same genes, yet they function differently? Information being the same, why are they functioning differently? Your heart cell, your lung cell, your liver cell, your kidney cell, your bone, blood, brain, muscles, every cell, they behave very much differently. What is the reason behind it? Let's have a look, people. So if you look at a particular cell which does not have a function assigned, it has got all the avenues open when it can go on to develop muscle cells or fat cells or bone cells or blood cells, whatever. So that is the curiosity. What decides their functional diversity? Why so many cells in the body? And this is where a lot number of informations have been generated, and that includes one of the most important candidate that we are discussing today, called the micro RNA. People, micro RNA, like mRNA, is also a form of photocopy of the DNA only. But unlike mRNA, it is not something that goes translated into the protein. So what we saw here, that an mRNA is something that will be translated into protein, and miRNA does not. Then what does it do? People, miRNA is involved in the fine tuning of the expression of a gene. Now, just to give you a little more clarity, despite all the cells having the same, all cells having the same genes, they differ in the gene expression, meaning not all the genes are expressed by all the cells of the body. They are very selective, and that difference is done on two bases. One called as spatial, another called as temporal, meaning one with respect to the tissue and another with respect to the time. Try and understand. Your lung tissue, liver tissue, kidney tissue, skin tissue, nerve tissue, they will all choose different genes to express, and thereby they become that functional cell. And then also there are genes which will only express at certain time and not before or after that. Take the genes involved in giving you the reproductive ability, the genes associated with puberty and onset of puberty, and in particular in girls the end of it also, what you call as the menopause. All of these genes are regulated, and in that regulation, one of the key most element that we have are called micro RNA. Let's have a look quickly as to what is that supposed to mean. Imagine here is a cell, and imagine within the cell the nucleus and then the DNA. All of you already know that the DNA has those dedicated segments called the genes. What do you call as the gene? The segment which is coding for the specific protein. And then those areas which are not coding for proteins called as the non-coding regions. You see, or what you earlier termed as the junk region. So what is demarcated by this? Now let's say here is a gene which gets itself photocopied, and if it gets photocopied, what will it produce? It will produce the mRNA. Remember, unlike DNA, which is made up of two strands zipped up into each other, the RNA is made up of single strand. So there is only one strand. Assume this as one of the strand of the zip. So in case you somehow arrange for another strand, they will zip up again as well. But then what happens? Normally what happens is that this RNA, the information which is there in such sequences of bases are read, and that information is used to convert that information or translate it into the language of protein. Proteins are the polymers of amino acid. So protein is made by attaching many amino acids together. But then think of a scenario where the cell does not need to express this gene. And despite that, if the mRNA has been created, what it will do is like stop the press, or to stop the RNA from undergoing translation. And how would it do that? The idea is simple. People from these non-coding or the junk regions are also transcribed or photocopied a unique form of RNA, what we call as the micro RNA, which is the celebrity of the time given their Nobel Prize significance. You see, what are micro RNA? People, micro RNA are the single-stranded structure or single-stranded RNA nearly, and that could be again a part of the exam's question, nearly 19 to 22 bases long. So if you count the number of bases, it will be nearly 19 to 22 bases long. They have partial complementarity. What it means, people, if you consider this as a strand, mRNA as a strand, this miRNA will have its bases partially complementary to the RNA here. This goes again with one understanding that you might have already developed, that the two strands of DNA, they always zip up in a sequence complementary manner. Only when they have the sequences which is complementary will they zip up. So remember, the miRNA has the sequence which is partially complementary to an mRNA, meaning it will zip up only in that region of complementarity. And the moment that miRNA zips up, it will make this mRNA unfit for undergoing translation. And that way, this mRNA would be broken down within the cells or digested within the cells by the dedicated enzymes. And these enzymes are nothing but RNase enzymes. So they will chop it down and prevent it from expressing. So what we have to understand, what these people have discovered, is that these miRNA produced from the junk region and also from within the gene's non-coding region. We will not get into that, it will not be very much important for the exam, but just know there are some non-coding regions within the gene also. But then non-coding regions and these miRNA, by binding to the mRNA, prevent its translation. Thereby they prevent the gene from ultimately showing up as protein. Now, did you note that this miRNA from this region was complementary to this? Just visualize the remaining part of the mRNA. It might have a sequence that will be complementary to another mRNA's sequence. So what you have to remember, and that could be again one of the part of the exam's question, same miRNA, same micro RNA can bind with many different mRNAs of different genes. And this is one of the major tools, people, that the cells use to prevent those genes from expressing which they don't require. And then if you have to understand it little deeper from the application perspective, try and understand. If there is any problem in the regions which is producing miRNA, it will lead to genetic anomalies or problems. And hence, any problem associated with the miRNA has been found to be connected with many genetic diseases. Primarily, you can say cancer or cardiovascular diseases, CVDs, or also found to be connected with Alzheimer's or many metabolic disorders. The problems associated with the body's biochemical pathways or metabolism. All of these disorders could be the result of faulty gene expression and hence could be found to be connected with the miRNA's overexpression or underexpression. So what we have to now understand through all this discussion, people, that miRNA is a magic master keys that the cells have through which they are able to control the respective gene expressions in the different cells. And it is this relative difference in the gene expression of the different cells of your body, based on what genes do they choose to express, you can understand the particular cell will go on to become a particular cell type. And other than explaining you the functional diversity, as I have already hinted, that yes, study of these miRNA hence helps understanding so many different disorders, cardiovascular disorders, inflammatory disorders, neurodegenerative disease, metabolic disorders. And now connect with that, not only the understanding of the diseases, people, but also we are now going to be using the miRNA which are lab produced. So the idea is today the mankind is capable of producing novel RNAs in the lab itself. That is called polynucleotide synthesis. So can we now, taking the leaf from the nature's book, design the specific mRNA, and that mRNA will be introduced into the cell to particularly bind to the specific mRNA that you don't wish to go on to protein synthesis. And that is how miRNAs could be used as the tool for therapy. There are a number of miRNA candidates which are being today considered, and they are in the different phases of clinical trials for the treatment. Again, you can remember that is where the question could be centered around the different diseases. The metabolic disorders, the cardiovascular diseases, the cancer, the inflammatory autoimmune diseases, neurodegenerative diseases, and even people, infectious diseases. If we can control the gene expression of the infecting agents, the pathogens, we can control their growth as well. So that is one of the major area of application that their finding is leading us to. Further, with the plants also, you can just give a quick glance, and if at all that is there in the different options in your objective question, that same miRNA based technology could also be used for various different traits that you wish for in the plants. For example, you wanted to have the various abiotic stress tolerance, thermal tolerance, the aquatic tolerance, or the pH tolerance, and so on. The biotic stress tolerance, the weeds, the pests, you can design the agents that could be harmful for the weeds and pests in particular. Take a development of male sterile plants, the seedless fruit development, right? The modulation of flower and scent, so you can have the different colors in the flower, quite different from their natural ones. Or even you can modulate their fragrances. How about the rose smelling like jasmine? All of this could also be done by controlling the gene expression mediated by the tools like lab generated microRNA. Now coming to what was that initial slide wherein I talked about the similarity between the two Nobel Prizes. Now you see, these people, Victor Ambrose and Gary Ruvkun, have been awarded the Nobel for discovering the role of microRNA in the gene expression. But at the same time, we can also have another type of RNA called siRNA, or small interfering RNA. Remember, this now will get more potential because UPSC will try to create some tricky questions for you. And these people have already got the award Nobel Prize in 2006 for discovering a similar type of RNA called siRNA. Just a quick, simple note on that. Unlike the miRNA people, which is produced from within your genome, that is an endogenous thing. If you can, through a virus, if the virus can take its RNA, by the way, you have to remember that the siRNA is double-stranded RNA. Please remember, even if it looks double-stranded, you're not going to confuse it with the DNA. It is double-stranded RNA compared to miRNA, which was single-stranded. Remember, that could be one potential area for question. So what I'm saying is, either the virus transfers the double-stranded RNA inside, or you can, in the lab designed vector is nothing but a carrier. So any capsule in which you have packaged the double-stranded RNA, both of these, even the virus has the natural ability to transfer, both of these can transfer their double-stranded RNA called small interfering RNA to enter into this, and in exactly the same manner, just as the miRNA did, just that before they bind to the mRNA, siRNA needs to be unzipped. These two needs to be unzipped. They will be binding to the mRNA and preventing its translation. Now, what you have to remember, and that could be the question, siRNA, unlike miRNA, which is, or microRNA, which is produced endogenously, siRNA enters into the cell from outside, either through the virus or made in the lab, called exogenous introduction. So what you now have to understand, people, that we can generate siRNA in the lab as well and transfer it into the targeted cell and then make that siRNA zip up with the mRNA, thereby preventing its translation, and then connect it with all the potential that we have seen already with the miRNA. This particular phenomenon, people, is what we call by different terms. And what is UPSC going to ask? Called RNA-IE technology or RNA interference technology or RNA interference. Why? Because we are interfering with the RNA and preventing the conversion of information into the protein. And third, it could also be called as gene silencing. Gene silencing. So you see, you're not allowing the genes to express and produce the protein, enhence the term. So remember, both these Nobel Prizes, and that is where it gets interesting, that they are both getting used for the same purpose. The only difference between these two Nobel Prizes is that what they have discovered is on a much bigger way the cell's own mechanism to control its gene expression. And taking a leaf from there, we can use that tool also to targeted gene silencing, just as what these people had discovered in 2006, but with exogenous RNA, which is coming from outside, called siRNA or small interfering RNA, for the same purposes. So much so that just remember, at this point in time, we have just a couple of images to. So can you see what is being shown here in the pink is the DNA. It is getting photocopied to produce the mRNA, and that process called transcription, and that mRNA is getting bound by the micro RNA. The moment micro RNA binds to it, this mRNA will become useless because it will not turn into the protein. Fine. So micro RNA is preventing its translation. So just to give you a hint as to how important this could be, just remember, few of the siRNA based gene therapy, called gene therapy because they are controlling the gene expression, have already got approved. No need to remember any of it, just the concept that I'm talking about. There are some drugs like patisiran, givosiran, and then inclisiran. Siran is because all have siRNA and that's the name. So they already have got approved to be used as the drug for multiple diseases. For micro RNA, we cannot say that as of yet, because we are still, in the phases of clinical trials where the, trials are happening if they could be used for the same purpose. Just a quick show of what we have discussed so far. So can you see the central dogma, which is the same story happening in all the life? The DNA photocopied, the RNA comes, and the RNA normally gets translated to form the protein. But then imagine what the RNA interference mechanism holds. The double-stranded RNA, this is siRNA, comes, it has been, unzipped, and this yellow color thing binds to the mRNA, and because of this, the mRNA is chopped down. No protein synthesis that will occur. Interestingly, just an information, even in India, the Drug Controller General of India had granted permission for India's first small interference RNA drug trial in 2016 itself. Yet to find any report on what happened to the clinical trials, but it was called QPI 1007. It was for an eye-related disorder. But that is siRNA. The present Nobel Prize is on micro RNA, don't make a mistake. Right. So there are multiple areas on which we have seen that therapeutic potential could be exploited. But then in case the question is around what are the issues associated, what are the challenges if at all associated with the micro RNA, you can just give a quick read, because if you are introducing any lab-made miRNA targeting a certain gene within few of the cells, it might have multiple toxicities. I mean, it's a repeat. I think the adverse effects, it may lead to low efficacy at high dosage. When you give too high a dosage of the micro RNA, it may also impact the efficacy of it to be used for the gene silencing. immunogenicity is another big problem, because if you are introducing a foreign microRNA, it may also be responded to through the immunological high excesses. And then efficiency of delivery mechanism or delivery system. How efficiently are you able to transfer the microRNA into the cells? So these are some of the aspect about it. Just before we end, a couple of interesting things about the Nobel Prize winners this year. You see, Mr. Gary Ruvkun, he got excited into the science more after the moon landing happened in the 60s, and then he was driven into it. But then interestingly, he went into the specialization in physics, biophysics to be particular. They have called BA in biophysics that he did, and then later he went on to study the molecular biology. So you can see the education system over there allows the creative thinking and interdisciplinary approach, wherein someone from physics is going on to win the Nobel in core biological sciences. And this is not just one, there are many. One particular aspect that UPSC may find to give you the surprise around the organism they have studied, people. To study all of this, the microRNA, they did not study initially in the humans, and that is why when they initially did it in 90s, it took over 30 years for us to even award them the Nobel. So the organism that they focused on is a very tiny little worm, a nematode, roundworm as you call it. Generally you associate with roundworms as a very negative relationship, as the parasites in our gut, intestine. But you see, these little roundworms, just a millimeter size, they over the decades have emerged to be one of the best model in genetic study. So model organism bulate isko, we call it the model organism. Yes, what is the name of this worm? Caenorhabditis elegans, or C. elegans it is popularly called. Now, there could be the question as to what is so special about this organism, so much so that 2006 Nobel Prize and earlier, Nobel Prizes also, and this year's have been around this particular organism. Just a quick look at why they are so useful. People, they are very tiny organisms, very easy to grow, in the labs, and they just require the aquatic medium. So they're very fast to grow, and just 2 weeks lifespan. So it is easy to, study the entire lifespan in a short frame of time. They are transparent, worms. So you can easily, under the microscope, see through the entire body and see the different parts, the organ system that is functioning. Their body is made up of just 959 cells. So you could remember, this, because they have just counted, and there is precise that much number of cells they have, not less or more. That, is something that makes them, the scientists, easy to detail study the organism and then possibly replicate it on the humans as well. And then they have been great model for aging studies. You see, I told you they have a lifespan of just 2 weeks, and that allows us to study the aging, which in the humans, let us say, happens in a much longer time frame, in a shorter time frame, and understand it better. And that particularly leads to the better aging related research. In particular, people, we should not ignore that we have also figured out that they have just 302 nerve cells, and we have developed the entire map of how these nerve cells are connected with each other, making what is called as the comprehensive connectivity map. Now, why am I mentioning this? Because by studying the different connections, synapses, the signaling, we are able to utilize the very same thing in also the artificial neural linking, what we are calling as the artificial intelligence. So these organisms have a strong influence on the development of artificial intelligence as well. So you see, and then the other Nobel Prize that we'll be talking about in another video, Nobel Prize in Chemistry, we will see how artificial intelligence has revolutionized the field of protein biology. So these organisms have a lot to offer, because based on the less number of neurons and able to find the connections and interconnections, and how they're able to analyze the information, we are able to develop the artificial neural linking as well. So yes, this organism has been one of the, I would say, biggest achievement for the mankind from the science perspective. And you can just see, it was around Caenorhabditis elegans that we got 2002 Nobel, 2006 Nobel, 2008, and this year's 2024. Tiny organism, we are so much indebted to. Before I just end this talk, quickly let us have a look at a beautiful video, a tiny little clip of Gary Ruvkun making a statement after his Nobel to some of his students. I just felt that it could have a strong message for the UPSC aspirants who many a times invite anxiety, not getting very clear-headed. Just have a look. Mr. Gary.
Yeah, it's been a good morning. Thank you. From like 21 to 24, I was a disappointment to anybody who was thinking about where my career might go, because I lived in my van and planted trees in the Pacific Northwest and traveled in third-class buses in Cochabamba, Bolivia. I was bored with travel, and I spent a day reading Scientific American, and it was really a good day, and I said, you know what, maybe I'll go to graduate school. So I came to Harvard for graduate school, and that was 76. That was sort of the moment when recombinant DNA was really just starting to take off. And it was obvious that that was a revolution. It was a sea change, and you could see that it dramatically changes the science. We run into this constantly in our little worms where people say, in nematode, it's not even a mammal. Right, and it's a slimy, it's a worm that's a millimeter long, But if you look at the animals that have the super potent small RNA system that worms have, it's scorpions and ticks and coral and clams. And it's the most badass organisms on the planet, right? And that makes our little worms badass. And no one ever thought to use that term for a worm. And so, we are asserting ourselves now.
I hope you understood that many a times not thinking too much is possibly the way to allow the right thoughts to come into the head. See you soon in another video. Thank you. Have a good day.